Without further ado, it's my real pleasure to introduce Ivan Soltesh, who's the James Doty Professor in Neurosurgery and Neurosciences at Stanford. I have read his papers for many years. This is the first time I've actually had the pleasure of meeting him. He does this elegant combination of physiology and anatomy and computational neuroscience, and the papers are just elegant and rigorous, and I can't think of a better person to introduce us to the circuits related to epilepsy. So, Ivan, it's a pleasure to have you here.
Thank you very much for this kind introduction, and thank you also for staying. I think we have a very exciting session, and there'll be really two talks, one after another. My talk on the preclinical evidence on cannabinoids and epilepsy, followed by Amy's talk on the clinical data, or lack thereof. So, in a sense, these two talks will look at the smoke and peer into the abyss of that crater and look what's the fire like there, what's happening down there. So I don't have anything to declare, and these are the objectives. So we will review the mechanistic aspects of cannabinoid signaling as it relates to epilepsy. You heard some of them already, so I'm just going to really briefly revisit them and remind you of the key aspects. Then we will consider the preclinical evidence for cannabinoids in seizure control. What do we know? And evaluate some of the risk factors and confounds. So I'm really going to focus on the underlying neuronal circuits, the neuronal regulations in epilepsy. So just a few words about epilepsy. As a reminder, it's characterized, of course, by spontaneous recurrent seizures. Millions of people have it. Estimates range from 50 million to up to 65 million people worldwide. There are also comorbidities, so when people have epilepsy, it's not just the seizures that are a problem, but also the cognitive impairments and psychiatric disorders. And there's also a 10 times higher risk of sudden death than the general population. So, epilepsy is really a huge problem. Now, really, we should talk about not epilepsy, but epilepsies in the plural, because there are many types. For example, we have temporal epilepsy, many kinds of severe childhood epilepsies, post-traumatic epilepsy, and many other kinds of epilepsies. And many of these are treatment resistant. Up to 30% of the patients actually have no good treatment, so they are living with seizures of various frequencies. The treatment today consists of largely of anti-epileptic drugs or surgery, and both of these classes of treatments have their major side effects. So even if the seizures are fully or partially controlled, in many cases we have significant side effects. So we really need to do much more and develop new avenues for therapy. So what are the key properties of cannabinoid signaling as far as epilepsy is concerned? So you heard about this from the previous speakers, mostly from Dr. Mackey. Well, let me just remind you of some of them. So if this is a postsynaptic membrane here, and these are the presynaptic terminals, two inhibitory terminals are shown and one excitatory terminal. The excitatory terminal is going to release excitatory amino acid glutamate that will trigger endocannabinoid, or can trigger endocannabinoid release, that will cross the synapse backward. That's called the retrograde signaling, and it can bind to these cannabinoid receptors in purple and block further release. You'll see that this is a key part of how we think about cannabinoid action in epilepsy. The other part of it, though, is that in addition to inhibiting further glutamates or excitatory neurotransmitter release, cannabinoids, endocannabinoids, can also inhibit inhibitory neurotransmitter release, GABA release. So in other words, it can decrease the natural inhibitory system of the brain. So as far as epilepsy is concerned, right off the bat, you see that this is going to raise some complexities in the tissue because you have the control of excitatory and the inhibitory terminals in the normal brain. And then when you add an exocannabinoid, the outcome will be really a question of balance between the excitation and inhibition, of the effects on the excitatory and inhibitory systems in the brain. Now, the other thing that's very important is that not all inhibitory terminals express cannabinoids. So this is very important because this means that only some cells in the brain, and actually numerically they are very, very few. So these kind of cells are maybe 2 to 3% in the brain. So only some cells contain these receptors, but not all. And notice also, as this cartoon indicates, that the inhibitory terminals contain many more receptors than the excitatory terminals. But we learned something the hard way over the years, which is that the CB1 receptors numbers do not always and necessarily indicate or predict functional efficacy. So this was really at the synapse level. So just for one slide, let's step out a little bit, zoom out, and let's look at now a circuit. So a few years ago, we discovered that, for example, when you look at these cells that I showed you in the previous slide on the right, the cells that express, the inhibitory cells that express CB1 receptors, they can actually participate in highly specific neuronal networks. So it's not like these CCK or these are cholecystokinin positive, also expressing CB1 receptor type interneurons, inhibitory cells. They do not just innervate randomly all kinds of excitatory neurons. On the contrary, they highly specifically innervate one type of postsynaptic cell. This happens to be in the medial entorhinal cortex, but the whole point here is the general point, which is that CB1 regulation takes place in distinct information processing channels. So in this case, these red cells project to the contralateral entorhinal cortex, whereas these blue cells that form the perforant path project to the ipsilateral dentate gyrus. And as you see, these CB1 containing CCK positive interneurons specifically innervate one type of cell and almost completely avoid the other type. So the key here is that when a drug hits the brain, it's not going to act equally on every cell. It's going to act on very specific cell types, which then participate in very specific circuits. And obviously, this is important to know if you are trying to design therapies based on this. The other important point that you heard already from Ken, the first session, is that the synthesis of endocannabinoids is activity dependent. Obviously, during a seizure, you have depolarization of the postsynaptic membrane, and this depolarization is a major signal that can lead to the synthesis and release of substances like 2-AG that then can activate the CB1 receptors. So, for example, in the case of an inhibitory terminal, if you depolarize the postsynaptic cell, that's shown here, if you depolarize the postsynaptic cell and you measure the amount of neurotransmitter activation here on the postsynaptic membrane following the release of GABA, the inhibitory neurotransmitter, here at the synapse, what you see is that following depolarization here, because 2-AG is released and CB1 receptors are activated, that transiently inhibit transmitter release, you see this kind of effect that following a postsynaptic action potential burst or any kind of depolarization that's large enough is going to suppress inhibition. So this is something that we have to keep in mind, that if you have seizures, just the building mechanism of depolarization-induced suppression of inhibition that Ken already mentioned to you earlier, that in itself is going to decrease inhibition and allow further seizures to take place. Now, what about the excitatory side, the glutamatergic side? And I alluded to this, but I want to get back to it again, because I think this is really critical when we think about seizures and cannabinoids. So as this figure illustrates, at low levels of activity, the way we think about it is that glutamate will activate, for example, AMPA receptors. This is one type of excitatory receptors. Whereas whereas at heightened levels of activity, more glutamate will be released that will also activate the so-called metabotropic glutamate receptors, a particular subtype. And these metabotropic receptors, as the name suggests, they start an intracellular signaling pathway that involves various molecules like G protein related molecules like GQ11, phospholipase C beta, and Doug lipase alpha. And Doug lipase alpha will then synthesize and then synthesize 2-AG that will then cross the synaptic cleft and block further glutamate release. So this, if you want, is a built-in synaptic circuit dampener as proposed by István Katona, one of my collaborators in Budapest. And the reason why this is a very important point is that this is really a built-in mechanism by which the excitatory synapse can lower further glutamate release, further excitatory glutamate release upon heightened activity. And importantly, these molecules are situated on the, just around the synapse at what we call the perisynaptic machinery. And when you knock out members of this pathway that I mentioned, like mGluR5 or PLC beta or DGL alpha, you tend to get seizures. So this just shows you that this built-in circuit dampener is really important. Now, not surprisingly in a way, if you have this circuit dampener that I showed you and you act, you put on exogenously applied cannabinoid ligands and you just basically activate this circuit dampener artificially if you want, you can get, and typically you get, anti-convulsant effect. So in animal models of epilepsy, there is broad agreement that acute exogenous application of a CB1 agonist is typically, not always, but typically anti-convulsant. Now, and the reverse is also true. If you block this built-in circuit dampener with an antagonist, as is shown here on the slide, SR is a CB1 receptor antagonist. So if you block that, then the circuit dampener is not going to work properly and you can make the seizures worse. So in general, CB1 agonism is anticonvulsant because you basically help this system, the built-in circuit dampener, and an antagonist tend to be proconvulsant. Now, very importantly, as we think further about epilepsy, one has to say that in general, the seizures make the CB1 receptor system really altered. So the cannabinoid system in an epileptic brain is not the same as the cannabinoid system in the control brain. This is really a critical point. In general, what we and others have found is that the CB1 receptor numbers tend to increase on GABAergic terminals. So we are blocking now the GABA release even more. And on glutamatergic terminals, the CB1 receptors tend to be down. And this is actually, the one on the right is from human tissue. So you can see that, this actually happens in the human epileptic brain as well. And what this means is, in a sense, is that this circuit dampener doesn't work as well in the epileptic tissue. Now, what I just showed you up till now was really in a chronically epileptic tissue. So, like in the previous slide, in this patient, had non-sclerotic or sclerotic hippocampi, so went through many seizures, severe epilepsy. Now, what happens right after the first large seizure-like event, status epilepticus, or a head injury, or some precipitating event? There is broad agreement, I think I can say, that right after there's the first severe seizure, there is a rise in endocannabinoids. And again, we can explain that because anytime calcium levels rise in the cell, you are going to have a rise in endocannabinoids. And in this beautiful study, it has been shown that shortly after status epilepticus, there is a massive downregulation of CB1 receptors from GABAergic terminals. So the point is that this surge in endocannabinoids may lead to a downregulation of CB1 receptors. And there have been efforts trying to figure out how we could actually figure out whether this transient downregulation of CB1 receptors immediately after the precipitating event, whether that's actually important or not, and I don't have time to get into, but it's an interesting area of research, and it might actually point to novel ways of preventing the long-term appearance of epilepsy after a precipitating event, this process of insult leading to an epileptic brain, what we refer to as anti-epileptogenicity, which is something that epilepsy researchers all around the world are really, really interested in. Now, cannabidiol, of course, is another topic because that we should at least briefly cover. There's huge interest in cannabidiol and epilepsy, so let me just say a few words about it in terms of the mechanism, the signaling mechanism, and Amy will then cover the clinical evidence. So cannabidiol, as was already mentioned briefly, is really the definition of polypharmacy. There are more than 65 different molecular targets that have been reported, and as you see, many of them are enzymes, some are ion channels, and some are receptors and transporters as well. CBD in general doesn't directly act through the CB1 system in terms of epilepsy. At least that's what's believed, and I'll show you some evidence for that later on. But what is interesting, and it's important, and it's really we need to do something about, is that we really don't understand what the major targets are in terms of any anti-convulsant action of CBD. We don't know what the targets are. If you really think about it, you try to pick out some of the potential targets. I think many researchers in the field would agree that some of these targets may include the GPR55, which is a very interesting and novel receptor. It's one of the, it used to be an orphan receptor, a G protein coupled receptor 55. Now we actually know the endogenous ligand, it's believed. But the point is that GPR55, as well as some calcium channels and other types of molecules, may be involved in as potential targets with relevance for epilepsy in terms of seizure control. And several of these CBD targets are involved in intracellular regulation and modulation of glutamate release. So again, we are coming back to the importance of glutamate release regulation. On the right, you see something that I already referred to, which is that cannabidiol can have significant and protective effect against seizures. This is all, however, in what we call seizure threshold models. So this is, in the high, in a tens of milligram per kilogram range. You see that that cannabidiol is effective. This happens to be maximal electroshock. That basically it's you take a normal animal and you shock it and you get seizures, and you measure how much cannabidiol you need to protect against that. And what you can see is that this protective effect of cannabidiol appears to be insensitive to rimonabant, the CB1 antagonist. So it appears to be independent of the CB1 system, unlike what you see on the top, where a similar curve can be seen for WIN, which is a CB1 agonist, and the CB1 agonist is very sensitive to the CB1 antagonist in terms of its anti-seizure action. Now, in the remaining few minutes, let me just highlight some of the issues that are important in terms of CB1 and cannabidiol action and epilepsy. One of them is that there is some clinical data, although much more research is needed, that exogenous CB1 activation can alter normal network oscillations. Now, epilepsy and seizures are clearly a aphysiological and not wanted kind of oscillation. It's a rhythmic event, electrical event in the brain, but our brain is constantly producing normal oscillations. So, for example, when a mouse, for example, is running around and trying to find food, you get this, what they call theta oscillation. It's a relatively low frequency oscillation, about 5 to 10 hertz. And when you apply some CB1 agonist, what we know is that the theta rhythm gets smaller, and this theta oscillation is believed to really couple neurons into firing in very sharp temporal windows, and this kind of cell assembly or organization is disrupted when CB1 agonists are given. And there are other types of oscillations that may actually appear upon CB1 receptor activation, and I don't have time to get into, but the point is these natural rhythms can be very strongly modulated by CB1, and it could be that that underlies all the cognitive effects that you have heard about today. Two other points. One of them is that, as you heard from Ken McKee earlier, CB1 signaling plays key roles in development. There's really no doubt about it. For example, you see here that CB1 receptor positive axons are growing from the cortex, and they're meeting MEG lipase positive axons that are growing into the cortex, and they kind of form kind of handshake. So there's very intricate CB1 signaling going on that guides these axonal developments so that they find their proper targets in the proper numbers and they make proper connections. So therefore, it's not entirely surprising that if you now in utero or early neonatal brains, if you apply cannabis or CB1 agonists, you can actually have ectopic formation of synapses and mistargeting, like it is shown here for a type of interneuron that I already talked to you about, the CB1 receptor containing inhibitory cells. And people believe that cytoskeletal dynamics is a key molecular target for cannabis in development. It's just simply because when you have axons growing, the cytoskeleton, the intracellular architecture of the cells is the one that causes these intricate, dense kind of mechanisms that occur between the presynaptic and the postsynaptic sides, and they find their targets. It's really intracellular motors and molecular mechanisms that mediate that. Now, what about cannabidiol? There is very little data on the cannabidiol effect on development. There is a recent study that I found very interesting from 2015. Ken is actually an author on it, but the interesting thing is what this paper looked at, and this is really needed, that is they found that when you look at, for example, axonal growth, these are called philopodia, in developing axons, this is or growth cones in vitro, and what you see is that when you apply cannabidiol, you actually have these axonal tips, these growing axonal tips called the growth cones. They become structurally simpler, so the bottom line is they change, clearly change I think it was 300 nanomole cannabidiol, which is a little bit on the higher range, but not completely out of the range that you may see in a child that's on cannabidiol. And you can read that there are also changes in neural growth. This is all in vitro, and they also looked at axonal branching in vivo by injecting cannabidiol into the eye and then looking at what happens to the axons as they grow out. And just very briefly, I want to highlight two areas, just two more slides, that I think we should really increase research, and one in terms of epilepsy and cannabinoid and the CB1 system. One of them is that there is very strong evidence now that GABA release, the inhibitory neurotransmitter release, is strongly regulated by not just by activity-dependent release of the endocannabinoids, but also just by the ongoing endogenous or tonic activity of the receptors on the presynaptic terminals of these inhibitory neurons. And to give you one little taste of how big this regulation can be, this is an inhibitory synapse here, presynaptic inhibitory cell action potential, releases GABA, but you don't see anything until you put on the AM251, which is like rimonabant, an antagonist of the CB1 receptor, where you see that this silent synapse becomes a very, very loud synapse. So what happened was that in this situation, you didn't actually depolarize the postsynaptic cell, but the CB1 receptors were actually ongoing in their activity, constitutively active, and suppressing GABA release just by that, without any kind of activity in the postsynaptic cell. And as you can see, it massively regulates the probability of inhibitory neurotransmitter release. I don't have time to get into, but there is a lot of evidence that this kind of regulation of the probability of neurotransmitter release by the CB1 system is in fact undergoing very, very significant long-term changes in seizures as well as in autism. And then one more point, which is that, just to show you how much more research is needed, just very briefly, a recent paper from our lab showed that in addition to the presynaptic receptors that we all know about, and we discussed that they can be on glutamatergic as well as on inhibitory terminals, but these are all presynaptic receptors, there is actually a postsynaptic signaling pathway. This is very exciting because now we know a little bit about the signaling mechanism of this postsynaptic pathway. These are in the dendrites, and it involves these channels called HCN channels, which actually control all kinds of phenomena in the dendritic processes of neurons. And what is very interesting from the point of view of cannabinoids and epilepsy research is that this pathway seems to be entirely responsible for the learning and memory effects of cannabinoids. So if we take a mouse and knock out HCN1, CB1 agonists don't seem to affect learning and memory. So this is very exciting to me because it means that maybe it will be possible to selectively block the cognitive side effects of epilepsy, of THC, and maybe harness the positive effects of cannabinoid control on epilepsy in the future. And finally, I just want to show you that very interesting results from István Katona's lab now actually came up with a new technology applying super-resolution microscopy where we can actually quantify CB1 receptor numbers before and after THC. And you can see that this can be coupled to electrophysiological and anatomical investigations of the brain. This technology has great promise, I believe, for epilepsy research, where we can really look at controls and untreated and treated epileptic animals and maybe even human tissue. So to wrap up very quickly, I think in summary we can agree that CB1 signaling is highly cell type specific. It's very important to keep that in mind. It affects both select inhibitory and excitatory synapses. CB1 agonists and cannabidiol are typically anticonvulsant. CB1 numbers change in chronic epilepsy. This is really important. And seizures and injury, so precipitating seizures and precipitating injury, induces rapid, robust changes in endocannabinoid signaling. What is it that we still don't know? I believe, as I said, tonic control of neurotransmitter release in epilepsy is something we have to look at as a community. This postsynaptic novel cannabinoid pathway also is very important, and I think there'll be a tremendous interest in it. The identity of the CBD targets mediating anticonvulsant action is going to be looked at, and it's really something we need to know. And the long-term effects of CBD and THC on brain development needs to be better studied, paying close attention to the concentration that we use. And in general, then, the anti-epileptogenic actions of cannabinoids will also be important. And I just want to thank the decades-long support from NINDS and the people with whom I collaborate, and they were very important in my career. Thank you.
Okay, next we have Dr. Amy Brooks-Kale, who is Professor of Pediatrics, Neurology, and Pharmaceutical Sciences, and she's also the Chief and Ponzio Family Chair of Pediatric Neurology at the University of Colorado School of Medicine and the Children's Hospital in Colorado. As I said earlier, she is a world expert in pediatric epilepsy. She works with families with children with all kinds of pretty bad disorders, and being out there in Colorado has provided some interesting challenges that she's going to share with us.